Hello. Okay, hi everybody. I think it's a good time to start. And welcome to this session. I am Sumitabha Brahmachari. I'm a postdoc at CTPP Rice University in Houston. And today's tutorial is going to be about, the mix of some lectures on how we do maximum entropy approach to chromatin simulation modeling. We'll have some hands-on approach. We will try to run some simulations on the Google Colab, and I'll also explain what's going on behind it, and we'll try to do like in steps. In the beginning, I'll basically explain some of the ideas, and then we can move on to a more hands-on approach. Okay, and anytime you have questions, please feel free to raise your hand, ask questions. We will also, when we are running the collab, there will be TAs around the room. Antonio, Mateus, who'll assist you. Just raise your hand when we are doing that. So more details on that when we come. So basically, the agenda for today is to give you a flavor of how we run polymer simulations driven by, and then optimize the parameters of the simulation driven by high-C data sets to obtain various structural ensembles of chromosomes, polymer ensembles of chromosomes. And then, as the title of the workshop here there is function in it. So how do we go from structure to function? That's not something we will exclusively go into, and it's a way where we have to obtain the function from the structure, and I'll talk a little bit about, and there are many posters on how we are doing this. We will focus on this part where we are generating the structural ensembles, and I'll give you some flavor of how we can get at function from these ensembles. So these are sort of going to be our main objectives of running simulations and analyzing some ensembles and optimizing the parameters. And so we will be using the OpenMicron software. This has been really a collaborative effort. As I was showing here, all of these people have critically contributed and lots of hard work in developing this software. So we will be basically using that software, and you'll get familiar with how to use it for running simulations and optimizing various aspects of it. Okay, so I won't bore you too much with details of basics of chromosome, but it's good to be on the same page. It's early in the morning, we all are sort of aware of what the typical length scales are. Something to sort of notice is that dynamics is overdamped in this situation, so there is no mass, there is no inertia. So all the dynamics is sort of in the overdamped regime. So that's what we will be employing for all our polymer simulations. Again, and the way we think about chromosomes is, is slightly different, not very different, but slightly different from what we were doing in the other workshops here. Our polymer approach is really central. We really think of chromosomes as a contiguous segment, as a long polymer segment, and famously it has a lot of interesting double helical features at the very small length scales, but we will be focusing more on the length scales of tens to 50 hundreds of kilobases, where the chromosome can be really coarse-grained or thought of as like monomers or beads of a polymer, which contain lots of DNA and proteins and all these things together. And the typical dimension of these beads are going to be order of 100 nanometers. That's the sort of physical length scales that we will be using in this workshop. So this is sort of 1 unit of our chromosome polymer. Okay. So when I say polymer, there are some expectations from the statistical physics of polymers, right? That how a polymer should behave statistically, and so for a polymer of n segments, the end-to-end distance, or the typical size of the polymer, scales with the size of how many segments you have. So it's all of them are sort of positive scaling laws, so which makes sense. Longer polymer should be bigger. But then there are certain specific scalings that we expect from different kinds of polymers, like random walks versus if you include some sort of self-avoidance or not. And from that, we can order of magnitude estimate or get the scaling relationship of what's the probability of contact between two segments that are n monomers away. You can think of that in terms of random walk as what's the probability that a random walker will come back to the origin after n steps. And that, again, scales something like this for various kinds of polymers. And if you plot experimental data, and I'll come to a little bit, but I'm sure you are familiar with the scaling of contacts in high-C maps, it doesn't look like a single power law. Right? So it's not right. It's not like the chromosomes are a homogeneous polymer that one single power law can fit, but there is a lot of interesting features at different length scales, and one of the interesting things is that it's not a homogeneous polymer, and it doesn't have the sort of translational symmetry, because every sequence, we learned yesterday that there's a lot of information encoded in the sequence, so which part of the sequence you are, the structure can be very different, and what we assume in arriving in these sort of relationships is that the polymer is featureless. It has every part looks the same. That's not true, and this information comes from high-C experiments. This realization that chromosomes are not really randomly organized polymers. They're highly hierarchical. We have seen these kinds of maps in the last yesterday. Very quickly, I don't really need to tell you, but every point is basically a contact frequency between two regions of the genome. We saw how we can do this PCA, the workshop material, the thing we will run on Colab, will actually also compute this principal component analysis or eigen decomposition. This is the principal component of from the principal eigenvector of the eigen decomposition, which basically encodes of the main variation in the map. And if you see that there is a sort of checkerboard patterns, what this encodes is this checkerboard pattern. And there are other interesting features, is like, symmetric, it's highly non-random. That's one of the main features. That's what's one of the most striking features, is like there is this highly non-random, and there is this non-randomness has a cell type specificity, and all these things are very interesting things that we are still trying to understand of how different cell types are folded in different way. What are the underlying function of that? So those are still open questions. Another interesting thing that going to be important in the first part of the talk is if we look at the principal eigenvector, it turns out that this kind of organization is what is called the compartmentalized genome. So the two parts, if we call them A and B, and as we said yesterday, these two regions of the genome are sort of the A compartments like to stay in close proximity of other A compartments. So if you look at the high-C map, the A compartments have a higher overall probability of being near another A compartment. Similarly, B compartment have a higher probability of being near another B compartment, which leads to this kind of a compartmentalized architecture. And this has led to really this has been very useful for a lot of physical models based on copolymer models of the chromosome. And that's something we will be focusing on the first part of the talk. And this is again work that has been built over multiple years from multiple people. So the basic idea is that the chromosomes can be thought of as a block copolymer, and the underlying sort of energy function or underlying potential that we can simulate a chromosome. So the idea is that if you take a polymer and then simulate it under some potential, it should represent the structures that we see from high-C maps. And turns out that, parameters aside, that the functional form of this potential can be divided into these kinds of physics-based categories. One is the potential can be divided into a homopolymer term, but this is basically the fact that the chromosome is a polymer, so it has nearest neighbor connections and all these things. So we will look into the details of what these potentials are in a minute. But it's good to sort of have a bird's eye view of what we are doing. And then there is another part, which is the block phase separation part. And what this takes into account is if we consider the polymer as A and B, so A being blue and B being red, there is a certain intensity of interaction, certain affinity of interaction between A's and A's, which is denoted by epsilons here. And this controls sort of the principal eigenvector. This controls the checkerboard patterns of the high-C map. So if you just run a simulation with this phase separation of a polymer, you will recover the checkerboard pattern, but you will not recover everything. The other important ingredient of chromosomes is that it's locally crumpled. It's lengthwise compacted, which is encoded in this ideal chromosome term, and Basically, this is an effect of a steady-state loop extrusion. So the idea is that once you turn on this ideal chromosome term or potential, a polymer which is, this is entropically dominated polymer, which is fluctuating, will basically fold into something like, locally crumpled structure, which is the effect of steady-state loop extrusion or lengthwise compaction. And we have shown that this actually has also, this potential is controlled by SMC complexes. So you can tune this potential based on how active SMC complexes are and so on and so forth. So using this kind of a potential function and optimizing the parameters using maximum entropy approach. I'm not going into the optimization of parameters for this model, but we will come to optimization of parameters. But let's say we optimize the parameters to reproduce the high-C map. It turns out we can actually reproduce the high-C map pretty reliably. So these are experiment versus simulation. You can clearly see the checkerboard pattern, and this is the scaling relationship, and you can see the simulations actually can capture this kind of a scaling relationship. So this brings me to the first part of the tutorial. So what we will do in the first part of the tutorial is run a block copolymer simulation where we will implement this kind of a potential, and we'll not go through parameter optimization. We'll fix the parameters and see what we get. Can we still get similar checkerboard pattern? And we will use some of the parameters for the ideal chromosome that we take from one of the original publications, which were already optimized. So that's what is going to be the first task. So we use Langevin dynamics, and then use this potential on Langevin dynamics to see to run the simulation. So okay, let's go over to the Dropbox folder, and we can start with the blocks. Maybe I can just do it like. Okay. So this is our first, this block is the setup block, so you should be able to, it will take a few, still running. Okay, so I'm without GPU, so these, the OpenMicron software uses GPU acceleration, so you can do very fast simulations. So if you are lucky enough to connect with a GPU, or if you have a paid subscription, things will run much faster. Otherwise, in CPU, things will be much slow, but don't worry about it. It's you'll still get a feel of how to run these simulations, even though you're not going to be running it for a very long time, right? Okay. So while so everybody has started executing, is it executing? Yes, please. Okay.
It's in the instructions in the online version.
That's a good point. Where is
if you go to the Google Drive.
So go to Workshop Runs, and inside Workshop Runs there is a notebook called ChromosomeSims Maxent .ipynb. It's the notebook file. Did everybody actually? Workshop Runs. Workshop Runs. Yeah, it's inside Workshop Runs. And it's the only notebook inside Workshop Runs. Anybody having trouble finding the notebook? Yeah, please raise your hand if you have any kind of issue, and we'll take care of it. Okay, one note, if you see here, in this line, it says anything beyond Python 3, like 3.11.1, just delete that, make it equals to Python 3. Otherwise, it might cause problems. Okay, great. So if you face any issues, please raise hand, and we'll come to it. So then this is the utilities block, which has all the functions. Let's not go over it now. We'll come back to these functions as we are executing them and trying to understand what exactly they do. So let's just execute this and move on, and we'll come back to it. Okay, so the next step is we want to download a Hi-C dataset. That's going to be sort of one of our inputs in the simulation, because we want to do a simulation where to identify the blocks of the block copolymer, we need the first eigenvector, right? So what we would do in this is, if you want to run locally, there is this block that you can uncomment, and I provide this matrix. But here what we can do is just execute this block. What this will do is get a data set, a very high quality data set for chromosome 20 of the cell line GM12878 from human. And this is at a resolution of 100. So I'm using Hi-C straw, but you learned how to use cool tools, and so there are many different ways of getting the Hi-C data set. This is one of the ways. So Hi-C straw is from Aiden Lab. What we're doing is pretty simple. It's just importing the data. So because it's a very very large data set, this data set is like 30 gigabytes or something. The good thing is we are not downloading it. We are just streaming the data, and we are just streaming the part of the data that we need, which is what makes it very suitable for this kind of an application. So yeah, then you should get something like this. This is the size of our polymer, 631. So this is chromosome 20 when coarse grained at the resolution of 100 kilobases. You have about 631 beads. Okay. The next step is to plot. So why don't I do this just for, yeah. Okay, the next step is to plot the data. And there will be a couple of different things. So this is the raw data set that we have seen also yesterday. This is basically raw count. So every bin is basically the number of counts. In order to do the simulation, we need to do a little bit of a pre-processing. And the main pre-processing we are doing is to convert these counts into probabilities. That's one of the things, because we are in our simulations, we can compute probabilities, probability of contact. So that would be a good thing to actually quantitatively compare to the simulations with the experiments, so that we have both of them normalized. So that's one of the steps, is to normalize every bin with its row. So you can divide the sum of the row, so which is basically the total number of counts per bin. You can divide all the elements by that. So you're basically normalizing by the total number. And you need to do one additional step if need be, which is if your nearest neighbor so the first element from the diagonal, if it's not already the maximum or not already 1 after normalization, we need to put it to 1, or we need to normalize by the nearest neighbor. This is basically ensuring that the polymer integrity of our simulations is maintained, because in our simulations it's always a polymer. So the probability of being in contact with your nearest neighbor is always 1. And we want to sort of enforce that in the data, so we normalize accordingly so that the next nearest neighbor has a probability of 1. So when we plot the scaling map here, so this is raw counts, and as you can see, when after normalization, everything is probability here, and the nearest neighbor has a probability of contact equals 1. So everything is basically shifted so that your nearest neighbor has probability 1, and all the other contacts have a similar decay pattern. Okay, so I'm not going over this, but basically that's what we do. That's what this normalized Hi-C function does, and you can go over it. And now that you have all these AI tools, it's analyzing codes is so much easier. So if you're having issues later on, you can always use these kinds of tools to see what this particular function does. But that's what this normalized Hi-C function does, and we also learned this yesterday in Alexandra's session of how to do these normalizations and how to do this probability of contact scaling. So okay, so we will use this normalized map as our input for our simulations. So first step is to get the correlation matrix. So we do one additional step compared to what was done yesterday. It was just to take out the scaling of the map and then compute the eigen decomposition. You can do one more step of normalizing the matrix, of actually turning the matrix into a correlation matrix rather than just taking the difference. So the eigenvector doesn't change, the principal component doesn't change, but this is something we historically do. So that's what this block is. That's what this block is doing. It's basically taking the correlation matrix. It's just taking the correlation between points, and then we are doing eigen decomposition. What we find is the first eigenvector has about 37% of the weight, so it encodes about a third of the variation in the map, and we end up with a map that looks something like this, right? So this is the correlation map. There are elements which are missing because of the centromeres, and this is the principal eigenvector from which we obtained from this correlation map. And the reason we did the correlation is now it's normalized between -1 and 1. Instead of, if you don't do the correlation, it will not be normalized, but the eigenvector will not care because the eigenvector is immune to any constant shifts. So now we are just plotting it on top of the raw map, and you can see that a lot of this checkerboard pattern actually follows this eigenvector, the principal eigenvector. So the next step is we want to use this principal eigenvector to generate the sequence of our polymer. So in our polymer, we want to color the beads red and blue. And how do we color the beads red and blue? It's very simple. You can go through a loop and basically say, okay, this bead the first eigenvector is positive, color it blue versus color it red. That's what essentially is being done here in this, where we are generating the copolymer sequence from the principal eigenvector. So once so you can go check out this function. It basically takes the first eigenvector, asks for a file name where it stores. We want to store it into a file name because we want to pass it to the simulation block later on. So as you can see, the total length of the polymer is going to be 631, which is the same as the length of the high C map, dimension of the high C map. The bead types we have assigned A, B, and C. So A, B, and C is the missing values, so which is the centromere type bead. And this is a sample, so this is how the sequence looks. It's basically a list of A's and B's and C's. Okay. This is something we have incorporated the high C data set into the simulations. Now we will initialize the simulation object. Any technical issues running so far? All good? I see Mateos is explaining something. Is it good? Sorry? No. You are generating everything here. There is nothing you are getting from deposited files yet. Okay, let's see. Any previous version of it which doesn't have this block. So make sure you have this block. It's basically, you might find an error here because this program function is not finding this file because this folder doesn't exist, so it's unable to create this. So this is basically checking if the folder exists or not and creating the directory. So you might have an issue here, so it's an easy fix. So any questions so far? Okay. So now we are going to initiate the simulation object. So the simulation is like an object created. It's wrapped in, uses Python, wrapped in Python, but it uses the simulation engine of OpenMM, which is a versatile GPU accelerated package. So we create an object and then try to change its various characteristics, various methods. We have defined various methods to change its various attributes. So here is the first step is creating this simulation object, which is called sim. And then we have to set a platform. The way this code is written, it will automatically select the platform in your system. But if you want to manually hard code it and select a certain kind of platform, so these are the 4 available platforms for this. CUDA is usually the fastest. OpenCL is the next fastest. And then if you don't have anything else, CPU will always work. So we can. Oh, sure. Sorry. Good. Is it executing? The cell executes? All good? Anybody having any issues? Should I pause more? Should I move on? Move on? Good? Okay. I still see Matheus doing some troubleshooting. Matheus, all good? Okay. So, anytime any of the block gives you an error, just raise a hand quickly so that we resolve this before moving on. Okay, so this is initializing the object, selecting a platform. In my case, it's CPU. I don't have GPUs, but if you have GPUs, it should show CUDA. So I think you'll either see CUDA or CPU in a Colab. Now the next thing is basically selecting where to save your outputs, and then there is an initialization. So here we are using a sort of a random walk initialization for the polymer. It requires the sequence, so make sure you put the right address to the sequence of the sequence file, and then then just load the initial simulation. So it should just say chromosome simulation has been loaded, and the next step is to add the forces into the simulation of what kind of. So you saw this potential U, so we want to add this potential U so that all the forces derived from this potential, which is a part of the Langevin dynamics, so this grad U term, where are they coming from? So we have to specify them. So remember the homopolymer term. So this is basically one of the parts of the homopolymer term. We are adding harmonic bonds between nearest neighbors. So you can. So these are methods which are already defined within the simulation object. So you can go to OpenMicrom, and the link of the OpenMicrom is already the GitHub link, and the link to the documentation is already provided at the top of the notebook, so you can go and click there and see what these functions, how they are defined, and so on. But it's basically harmonic bonds, and this is the stiffness of the bond. You can also define other things like how far you want to make the rest length of the bond, and so on. Okay. The next thing is we are adding an angles potential. So what does this mean? This is basically controlling the local stiffness. So this is a 3-body term which controls the angle between 3 beads. So it controls the angle. So you can make it, if the angle is set to like 180 degrees, which is the default here, then you basically favor this configuration rather than a more kinked configuration. So you can choose either way, and this is the stiffness associated with this potential. The third one is the repulsive soft core potential. So this is non-neighboring beads which are not really bonded with each other. They feel the presence of the other bead. So this is sort of a self-avoidance, right? So that is modeled as a sigmoidal sort of a function, that if the beads come near a certain distance, in this case it's about 1.5 the diameter of the bead, then they start feeling a repulsive potential. So it's modeled as a sigmoidal curve. Okay, so it's soft core because the beads can eventually pass through each other, so it's not completely hardcore, and that's something that we want to incorporate in the simulations because of multiple reasons. One being, okay, It's motivated by biology because DNA segments can pass through each other because of DNA topoisomerase enzymes. It's an essential enzyme that's required, otherwise the cells malfunction. So, and the second more technical reason is if you put hardcore potentials, that things will not be able to equilibrate and will have a very hard time equilibrating in a certain landscape. So we add this softcore potential. The third one is it's a confining potential. It sort of mimics the nuclear confinement. It's not very essential, but it's usually good to sort of for the initial collapse of the polymer. You can later remove it, but in this case we will not. So it's basically a sort of a flat bottom confinement. So it's basically zero forces till about 15 sigma, 15 diameters, and then then there is a wall that starts harmonically. Okay. So this is sort of all the homopolymer parts. The second part is the block copolymer interactions. That's what we want to add. And as I mentioned, we have a while I'm explaining it, we should all execute this so that if there is an error in the meantime, we can fix it. Okay, so the third type is the type-to-type potential. So this is basically the interaction between A's and B's, or A's with other A's and B's with other B's, and so on. So you can imagine that there's going to be a 3x3 matrix, but it's symmetric because the way A interacts with B is the same as B interacts with A. And we are just defining this matrix. The more rigorous approach would be to find the values of this matrix from the Hi-C data sets, and that has been done, and these values are in the ballpark of what the optimization gives us. And basically that A's and B's interact, AB interaction is sort of similar as AA interaction, but BB interaction is very favored. If we assume that what I mean by B is a heterochromatin, then heterochromatin interacting with other heterochromatin is a stronger interaction than A types interacting with B types. Okay, so that's already encoded here because it's ABC, and this is B interacting with B, which is slightly higher than A interacting with A or A interacting with B. Okay. Okay, so this is sort of the types interaction matrix, and there are some steps to how to add this to the simulation. Then this is the part where we are adding it to the simulation. And then there is this ideal chromosome potential that I talked about, which is the steady state loop extrusion. And these parameters are sort of, imported from published data set, which, has gone through maximum entropy optimization to fit the data set. This parameter works, but it's not the only choice. You can tune the parameters and retrain, and you'll get slightly different set of parameters. So we will be using this one in absence of any new training that we'll be performing here. So these parameters are work for just running the simulations here. Okay. So now in this block, we will basically check what the forces are in our simulation. You can go deeper. So this force dict is the dictionary that contains all the forces. So you can actually go deeper and see what are the number of particles, what particle is this particle bonded to, and so on and so forth. You can really investigate what the forces are added. But generally, the keys of this dictionary is basically giving you all the potentials that are already in the system. Okay, so we have added all these potentials. Okay, great. Now our simulation setup is ready. We want to just run a collapse simulation so that we generate an initial configuration. From then we can start to sample our simulation distribution. So because I have GPUs, I'll run less number of blocks. But if you have because I have CPUs, I'm running less. But if you have GPUs, you can run for more. What this is basically it runs 2 blocks of simulation. Where within each block you have 10,000. We have 1,000 steps. So these are the 2 numbers basically, 2 and 1,000. So it basically you saw that it ran 2 blocks, that added the forces. It gave this output that added the forces. These are the lines for every block that it executes. What you can see is that it gives you sort of a kinetic energy, it gives you a potential energy, the radius of gyration of the entire system. So clearly, it's still going down. The radius of gyration is still going down, so means the collapse has not really happened very efficiently. And so for that, we need to run for a longer time. So I'm not doing that. But if you have GPUs, you should definitely, because you see, this is SPS is the steps per second. So this is a sort of a determiner of how, indicator for what's the speed. If you using CUDA, you should see it about 20,000. Whereas in CPUs, it's like 100, a factor of 100 slower. Okay, so after this block, we have the simulation set up, all the forces added, and collapsed under, so running some initial simulation, so it has collapsed and given us hopefully a good initial configuration. So we can now start to store data set, store the simulation trajectory for a downstream analysis later on. That's what's being done here. It's basically you need to sort of initialize a storage, and then we are running these simulations again, because I don't have that many. So I will only run, let's say, 30 blocks, okay? So you should execute this block, and, okay, I see. So okay, it's important to know what this temper polymer block is doing. What this is basically doing is heating up the polymer with some temperature, and then it cools down. It's just to randomize the initial position. So what you can do is, if you really want to run for a multiple replicas, which is what is advised to do for if you want to get a lot of good statistics, then you can increase the number of replicas, and for every replica you are heating up the polymer and then collapsing it back, so which gives you enhanced sampling of the entire landscape. This is basically a way to get enhanced sampling. This is running very, very slow. So because the inside temper polymer, my steps of how many steps it should run is already defined. So what I will do now is for making it run faster, I hope it doesn't crash. Okay, it didn't. I'll turn it off. Okay, so I'm not doing the tempering part. So, follow the same thing that I did. If you have CPUs and you don't want to run for a very long time, don't rerun this block. Otherwise, it might ask you to actually restart the entire simulation object. It will give you trouble with that. But then you should be able to run this, and this is basically running the simulation block and saving the structure, and then finally closing the file. Okay. So let's wait for this to finish, and anybody having any issues, and if this is not happening for anybody. It's working? Everybody is working? Okay, great. So in the meantime, we can take a quick look at what is being done at temper polymer, just so that, because we have some time here. Yeah, so this is the temper polymer block. It's, the highest temperature is 400, and we are running this at 120. So it gets the temperature of the integrator, and then divides it. So my DT step is 30, so you can all define this. Then finding how many steps do I need to go from 400 to 120, which is the actual temperature of the simulation. And then basically run the simulation and change the temperature of the integrator in step by step. And then finally, when you have reached the final temperature, you run some more steps just to equilibrate it better at that temperature. So this is a very simple way to heat up the polymer so that you can do enhanced sampling. Okay. Is this block done for everybody? It's done for me. Yeah. Still running for anybody? No? Okay. So the next step is hopefully what you will see, okay, now it's time to see this, what has been created. So you will see because we defined copolymer output as the save folder in your top here. We defined want to save this inside a save folder. So inside a copolymer output here, you will see that there is a CNDB file that is being generated. Okay, so this is a binary file format that has been that is basically based off of sort of container style storage of HDF5 files, and so it should have all the trajectory information of all the beads basically at different time points. Okay, so you want this file, so this has all the trajectory information, and we can analyze the structures from this file. So this file should have been generated, and make sure that you have this this storage closed, because if you don't close the file, it will show as corrupt. Okay, so now the next step is to load the trajectory. So we have CNDB tools, which is sort of a tool set that analyzes the CNDB files and also does many other things, and we'll show some of them here. But yeah, so here basically you load the trajectory, get the XYZ of the trajectory, and then we can calculate what the simulated Hi-C map should look like from here. Now I have very little data set, so it would be only 30 steps, so it will basically look like a single cell Hi-C, so yeah, so this is how it looks like. It's very less data. It doesn't have any of the block patterns because we didn't run for a longer time. But if you have GPUs, I'm pretty sure, and if you have run for the default ones without changing, I'm pretty sure you'll have a little better coverage of the Hi-C map. And this is the scaling. Scaling is off, and if you run for 1,000 steps, you'll get a good scaling at least. If not the whole block copolymer picture, the scaling you will get pretty good. So, is everybody able to sort of run this and get this, run this block and get some comparison of simulation? So this is a comparison of simulation versus Hi-C, as you can probably make out here. The bottom one is the input data. The top one is the output data. This is again experiment versus simulation. This looks terrible because it's not sampled well enough. But if you do sample, how does it look? We will do that. But we can do multiple other things, right? We can look at the structures. We can analyze the structures. So this is another way to sort of analyze the structures, where we can get the positions specific to A, B, or C beads. So that's what is being done here. And then we can compute the radial number density across, from the center of mass of the polymer, and then we can plot. And this is something that we get is the C beads are inside, the B beads are basically more just outside the C beads, and A beads are more peripheral. So this is something you can just this is where you start to think about function. Is now that you have obtained the structure, we analyze the structure, and then we start to think about function. Why are A beads outside? What does it mean in terms of the function of the chromosome, and in what scenarios does it change, and what does it imply for the biology of the system? So that's when you can start and probe the functional aspect of these structural ensembles. Again, that's something. There is no one-shot way of doing it. So it's very problem dependent, and I'll talk about one aspect in which you can sort of get at that. But go through the posters. There are multiple posters where this problem is being tackled from various approaches. We can also calculate the dynamics. So Simon yesterday showed us very nice way to calculate MSDs from experimental data. We can do the same. So this is compute MSD. We can load the trajectories. This was done for a very long time step. It will be terrible. For this, for ABC, this is the mean square displacement. I don't have many points. And this is for a power law of 0.5. You can see that the subdiffusive power law already is pretty apparent from this, and this basically comes from the polymer nature that you would expect a subdiffusive power law. For any monomer to move, you have to drag your buddies, drag the chain along with you, so which stops you from being diffusive, which makes your motion more dampened and more subdiffusive. So hopefully you were able to run all of this and generate all these curves. So there is something which is okay. I provided in longer runs, there are some trajectories which are from 40 replicas run over 100,000 time steps, so it's well sampled, and you have to upload it into the Google Drive. We are not doing that now, and from here, I'm just going to show it to you. This is what the well sampled trajectories look like. So what you can see is that there is a lot of fine features in the Hi-C map, experimental Hi-C map, that is not captured. And that is not captured because what the input of the simulation is the principal component. So what we are aiming to capture here is basically the red region should show red region. Okay, so this is flipped. Remember the comment yesterday of how A's and B's can be flipped by there's an arbitrary sign. So I never noticed this before, but blue and red should be compared. One should be flipped with respect to the other. But what we are basically after here is the sign of is the relative changes between A and B compartments. Do we actually capture that? Because that's what the input of our simulation was, and we can see that many of these blocks which show high intensity here, low intensity here, those aspects are captured in these simulations, and the scaling is also well captured in these simulations. If you want to do better, if you want to actually capture all these fine lines, one trick to do this, in case of mammalian chromosomes where you still see this checkerboard pattern, you can increase the number of types. So instead of ABC, you can make it 6, A1, A2, B1, B2, B3, and that will start to make these maps more like the experimental maps. Okay, and go through and that has been done and shown that usually 6 types of beads is very good to capture most of this Hi-C features. Okay, so we are at the 1 hour mark, and the next step is going to be going beyond these copolymer models. So this was the first part of the tutorial. Any questions so far? Any technical difficulties running this? Any questions, technical questions or otherwise? I'll go back to the slides for some time, and then we'll come back to running this again. Good? No comments? Okay, let's start. So this is what we did. We ran simulations, structural ensembles, analyzed structure, and we got some analysis done, and we compared the Hi-C data sets with the simulations. Okay, the next step is sort of going beyond these copolymer models. The copolymer models work very well when the Hi-C structure shows a very checkerboard-like pattern, which is very evident that a copolymer model can well capture these kinds of principal components because it looks very checkered. But if you go to some other organisms, for example, this is Bacillus subtilis. This is a rod-shaped bacteria. It's a model organism, very well characterized. And this is the Hi-C map of its only chromosomes. It has one singular circular chromosome. You don't see this checkerboard pattern, right? So a copolymer model is not going to be a good base model to start with for this kind of a system. So for this kind of a system, we need to make our model much more generalized. We have to go beyond this sort of a copolymer approach. And that's what has been developed more recently at the center. And so let's rethink the problem a little bit and think about how do we do this. So what's the physics of the problem basically states that if you have a polymer configuration, if you have an ensemble of polymer configurations, which is denoted by this psi here, then we can, for a polymer, we can write what the sort of partition function is, because this partition function is basically the weighted, the sum of these exponential Boltzmann factors, right? And from there, given this partition function, we can compute what's the probability of contact between any two pairs of loci is. So let's say I and j, we can define a contact function that looks like this, which is like sort of a sigmoidal function. So when they come close with respect to some cutoff, we are not really cutoff, but the parameter, we call it a contact. Otherwise, we call it not a contact. Then we can compute what the average contact probability is between I and j once we have this kind of an ensemble. And by doing this, now, as soon as this average that we find is equal to the average that we see in experiment, we can be sure that these ensembles, structural ensembles, are representative of what we see in experiments, right? So our task here is to find the right potential so that the phi ij's that we compute from simulation is equal to the phi ij's that we compute, that we see in experiments, right? And this task was done in case of copolymer models, where so the most general form is this kind of a form, right? The potential can be divided into a polymer part, because that's our null hypothesis, that chromosome is a polymer, it always remains a polymer. And then on top of it, just a polymer is not going to be enough. We have to add some bias to it. And what I showed you before is that the bias can be, you can physically argue when you see it's more sort of data driven, and that where you can physically see that it looks like a copolymer. So I can modify my bias into a more generic terms that look like a copolymer, and then sort of characterize the bias. But when you don't see these features, you want to tackle this bias in a more generic form, right? So what's the most generic way to do this is through the principle. So we use the principle of maximum entropy, which was proposed long ago, and for everybody, especially the graduate students, I highly recommend reading this paper. It's a fantastic paper on doing this inverse statistical mechanics. The idea is to sort of find the right probability distribution given us, and then from the probability distribution, assuming that there is a equilibrium distribution, we can compute what the potential is, right? And the probability distribution that is correct is basically the one that maximizes the entropy of the system. And I find this line very interesting and very informative. What it's basically telling us is, like, if you go through this approach and your results don't agree with experiments, doesn't necessarily mean your model is wrong. What it means is that you have not incorporated all the constraints in the experiment. So I find that very insightful when doing these kinds of modeling. Okay, so doing the constrained maximization of entropy, we find that the most generic form of bias potential, without any underlying assumptions about copolymer or anything else, is this kind of a form, where it has the potentials are, the contact probabilities are sort of there are these Lagrange multipliers that are added to these contact probabilities. From here, we define an objective function, the only requirement being that it has to be convex so that we can find a minima, which has this form of a free energy. And as you can see, by choosing this convex objective function, the gradient of the objective function is already giving you what we want, that how phi ij experiments are comparing with the simulation, with the obtained simulation averaged phi ij's. Right, so we devised this kind of a iterative update where we run the simulation, we compute what your simulated phi ij's are, and we saw how to do this. So we have already seen this part. What we are going to do next is we're going to define a potential which looks like this. We're going to do this part. Then we're going to compute this gradient, which will take into account experimental high C maps, and then sort of do an iterative update of these biases following this optimization. Yesterday, Professor Fudenberg mentioned this Adam optimizer. So we are using basically the Adam optimizer. We've implemented a form of the Adam optimizer for this iterative update. And so you see that we don't incorporate the high C at the very beginning, which is what we were doing in the previous case. We were taking the sequence from the high C and then running the simulation. Here it doesn't matter where you start the simulations from. It's a random initialization. And then when we are doing the iterative update, we want to get the information from the experiments to basically decide what is the update that we want to do, which is the direction that we want to push these all these different lambdas, so that we can converge to a map which is close to the simulations. So this is the general approach. And when you do the optimization, you should have a. So when you do the optimization, this is I'm showing you all the showing you a run, and unfortunately we will not be able to do this run in a realistic time of minutes. This will take multiple hours of GPU simulations. But what you can see is gradually the error is decreasing, and with number of iterations. And then it sort of saturates, and you can see that this is the lambdas, the optimized force field, that we get some optimized force field, and this is the simulated contact map that converges to the experimental map, and this is the. So if you run this for long enough, we can do this. But for now, we are going to run sort of get you a feel of this, where we're going to run these simulations with polymer ensembles, simulation with polymer potentials to generate structural ensembles, but we'll also loop through this parameter optimization step. So everything is same. We're still doing Langevin dynamics, and there was a question about Langevin dynamics, and I think it's a very interesting, important question to address. So this simulation package is modular enough that you can use any kind of integrator. Langevin dynamics is the one that we use ports from OpenMM, which is what the OpenMicrom is built on. But you can write your own integrators and plug it in, or you can use any other integrator that OpenMM ports as well. So it's modular in the sense you can actually, the dynamics is not limited to this. You can actually change the dynamics according to as you want it. And I say this because you might want to think about beyond equilibrium, like activity and things like where forces are applied. So those things can be done and has been done to incorporate within this framework. Okay, so we are going to use this potential with this form to look at this map. So back to the Colab notebook. Okay, so this is what we call this agnostic learning. So we're learning the potential without any underlying assumption other than whatever we decide to be the null hypothesis. Here, with the null hypothesis for us is the polymer. You can define a different null hypothesis and still do the same procedure to learn. So we start with this. We get the data from this source. I would ask you that this is by default. This is basically showing. It's getting the data from a local repository. So I would ask you to comment the last two lines out and uncomment the first block of the code. Okay, so that and save the file to the local repository, to the local directory before running this block, and then it will basically download the data from the source rather than accessing your local repository. You can choose to upload your data, upload the data presented in the file, or you can just unblock, load this block. And what you will see is after this, although it's not necessary, but you will see this text appear here. And what is being done in this part is basically the same normalization that I talked about earlier. Is the nearest neighbor normalization that has already been done in this part of the code. Okay. So after this, we will be able to plot the data that we just got, and this is the results for the data and the scaling. And so I should stop here and make sure that everybody is on board before proceeding forward. So this is, again, I'll put it here. This is the block. I apologize about not providing you a properly commented block, but it should be simple enough. Is it Is it working? Okay, I see people looking at this code intently, so I think, yeah, I should probably stop here. You see, yeah, follow these lines. Just uncomment the thing within these lines, and delete the destination before that is mentioned before this file. If you're confused, you can just completely comment out the save text. It still works because we are loading the map into this variable, and we'll be using this variable. It's just residue from what I was doing before. Okay. Anybody still having issues with running this code? Well, anybody having issues with running this code? All good? Proceed. Green lights. Okay. Great. So we plotted this data, and this is, as I showed, this was the data. It's a circular chromosome. So you expect the corners to be enriched because your furthest neighbors are actually in contact. So you expect these corners to be in contact because of the underlying 15 minutes? Okay. Should hurry up because of the underlying assumption, but then there are new features of this X, which basically says the two arms are sort of zippered together. So can we get something about can we learn the effective potential for this, right? We can do the same analysis that we did before. The eigenvector weight is comparable. You can do the same kind of analysis, and this is what I was showing you before. Is you can see that the block copolymer is not a good null hypothesis to start with. So we are doing this a more agnostic approach, where every bead interaction is basically being learned. So we are trying to learn instead of, probably I should have said this before, where instead of averaging blocks and trying to learn the interaction between blocks, we're trying to run every pairwise interaction between every blocks in this simulation. That's what is basically the most agnostic approach is what we are implementing. Okay. So in order to do this, we have a different object, which is the initialization object, optimization object. So we, okay. Which version of file is this? Okay. So make sure you give the right path to the Hi-C file. Just delete the trailing strings. So we are initializing the output object, initializing the optimization object, which takes in the Hi-C file and basically computes the gradient and gives the updates to the lambdas as we go through. So the initial force field is stored in this, and it's basically zeros. So we are starting the initialization with all zeros, and you can choose to do a different one. Here we are initializing the simulation object. And and it has a underlying step of collapse step, which is being executed right now. Okay. And so it's basically being executed. The collapse step and its the initialized simulation is basically the same as the one we did before. Here we're adding all the forces. Let me just show you while it's being executed. We are adding all the forces, and where is it? Here. So we are creating this object. We are selecting the platform. We are adding all the forces to the system. We have added a cylindrical confinement because of how bacterial chromosomes are. It's cylindrical. And then we are doing a collapse simulation and then initializing the storage, and so on. So this was the initialized simulation part. The optimization basically is another object that is being optimized. It's called Adam training. We are using Adam optimizer, as I told you before. We are setting all these experimental high-C data sets, and then we are generating a sequence. Then we are sort of uploading, we are initializing the force field matrix with zeros. You can choose to initialize with something else. And then we are basically creating the first lambdas. And then in the train part, what we will do is our training involves an iterative step. We have n number of steps. Then, for replicas in this range, we will, do the tempering of the polymer, and then run simulation blocks. Then we want to update the parameters, which is what the lambda updates are. So this update parameter has the update equation, and then we want to update this force field of the simulation, right? Because the new lambdas need to be incorporated into the force field and run new simulations to get the new data set. Okay, I am going to comment out the temper polymer part because I have CPUs here. So now after the simulation is being initialized, I want to run this train step. I'm doing 2 steps, 1 replica, that's to 30 blocks. It's very, 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 very sparse, not good data at all. But it will run, and I'm not doing even iteration. So it's just running, it's not going to give you good results. But if you have CUDA, you can run for a little longer, like 4 steps and a few more replicas. So then it basically iterates through all these blocks to find what so this iteration is basically running the simulation to calculate the average phi ij's, the simulation phi ij's. And then it basically removed the custom types, the type-to-type interactions, computed the updates, and then added back the new updated interactions. So this is the sort of the error from error between high-C and experiments. So this is sort of a normalized error showing that the error is huge, and which direction to propagate will depend on that. Okay, so it's still running the second iteration. It should stop after this. Okay, so it ran through 2 iterations. So the plot training is basically a bunch of code to do the plotting, and you can look at it at your spare time. But you can define your iteration here, and it will basically plot something like this. It's terrible. You don't have many data sets. So the optimized force field is all zeros. It didn't get enough time to really sample and update. But if you do longer runs, and this is what I have given in the longer runs folder in the optimization, you can go to something like iteration 75, and what you can see is this is the sort of force field under which you can get the bottom is simulation, the top is the bottom is experiment, the top is simulation, and you can get a fairly good agreement between simulation and then the next step is using these optimized force fields, we want to run more simulations. So you can just use the same kind of initialized simulation, but now the initial force field you can define it to be the new lambdas or the new force field that is being learned. So you can just import that and then run the simulations, and so that's basically. And after that, you can follow the same steps to analyze various trajectories, and then go towards more of a functional aspect. Okay. Was everybody able to run the train part? Because after that, it's again following the same process. You can load the trajectory, you can run the simulation again using the new force fields, and then do all sorts of analysis. And what analysis to do to find what is that's where the creativity comes in, because this is just a tool that you can implement to do various things. But how are you answering a biological question will really depend on what you want to analyze. What is the right thing you want to analyze in these structures, and that's really where the most cutting edge research is going to be done on these aspects, is to really look at the structures and analyze statistical things that are going to tell us more about the biological mechanisms. Okay, and so that is beyond the scope of hopefully you guys will tell me about it when we next meet, of how you use this to analyze a real biological sample and then got some results out of it. I'll give you one taste of it, which So what we did here is we ran it for a wild type cell, but since there are data available for mutants, we also ran it for various mutants, right? So this is Bacillus subtilis for wild type, and this is for, PAR-AB is a protein system. So there are two proteins, PAR-A and PAR-B. Depletion of PAR-AB is known to cause problems for chromosome segregation. But this is the Hi-C map, so we don't care about what PAR-AB does. We can just take the Hi-C map and then learn the structures from it. Similarly, we can do it for SMC complexes, another protein. So depleted SMC complexes, what you can see clearly is that the cross-shaped is gone. So what is the function of this cross-shaped structure, right? That's a question that we can ask. We can use, so this is another way is that I wanted to highlight to you is this approach only takes into account Hi-C datasets, which is pairwise contacts. And the structures are consistent with pairwise contacts. So we can use all the imaging data as sort of a validation for these structures because this model has never seen imaging data, right? So this is something what is being done here. Certain loci are being imaged, then we can sort of we can compare images to also, this can also be used as a step to calibrate distances in our system. And so what should be the bead size, optimal bead size, which is also consistent with the imaging data. So that's another step that we can use to calibrate with experiments. That's what's being done here. And then I'm not going to bore you with the details, but I invite you to the poster where basically what we are doing is from these structures, we are trying to understand what the underlying biological mechanism is for chromosome segregation, and how these protein mutants are segregation deficient, and that's something we can actually tease out of this mechanism from the structures. And there are multiple other posters from CTBP also implementing these approaches. Like Antonio's poster will give you more details about this structure. It has been a lot of hard work on his part to build this software. Similarly, Vinicius and Mateus, who have actively contributed to building this software, and they all have posters. So please stop by, and I guarantee you'll learn a lot. As well as all the others have contributed in many different ways. With that, I would like to thank you for your attention. Thank the funding sources, of course, and of course, thanks the organizers for giving me this opportunity to present, Ed and Alexandra. I've been in contact with them. They have been tirelessly working on setting up all these things. So thank you, and thank you for your attention. Any questions?
Can you just tell us a little bit about what goes into the ideal chromosome? It's like the very beginning here.
Sure. So the add ideal chromosome part is basically a potential that looks like this. Okay. So this is the distance between two beads on the x-axis, and this is the interaction strength. This is still a local potential. It's not a long-range potential. So only when two beads, two monomers, which come in contact, and you define contact using a sigmoid function, depending on the distance between them, not the physical distance, but the distance in the genomic polymer contour, basically the contour distance between them, there will be a force, adhesive force, that acts on them, which follows this kind of a pattern. So monomers which are close by will have a higher adhesive force compared to monomers which are far apart. So this is sort of thinking of loop extrusion in the sense that you have loop extruders binding and extruding loops. So shorter loops are going to be much more stable than longer loops, which is why you have this kind of a potential that if you are talking about two monomers which are very far apart, the interaction strength is much lower than if you are talking about monomers which are closer. And this is the generic form I'm showing. You can always tune it to match to certain cases. Like if you have depletion of SMC complexes, how does this change to capture that. And the generic effect that you see is, an entropy-dominated polymer starts to collapse into something like a lengthwise compacted or locally compact, a locally crumpled polymer, which, you can think of this as being renormalized into a thicker polymer, essentially, in some sense. So that's what this is. The functional form, the functional form that we are using has 1 over distances and 1 over distance squared. But it's important to note that that's not, the particular mathematical function is not important. As long as you have the functional form, you can always fit the parameters to find the equivalent ideal chromosome term. Does that answer?
Yes.
Okay, any more questions? Okay, so I look forward to you guys using this software, and and feel free to reach out to me, Antonio, Vinicius, anybody from the center if you have any issues with this. Thank you.